Namaskar and good morning. Welcome to CEC Gurukul Lectures. I am Dr. Shantanu from Department of Botany, Deshbandhu College. Today we are going to start the type studies of bryophytes. We will be discussing various genera which fall under bryophytes. To begin with, we will start with the group Hepaticopsida. Now, before we start our discussion towards various different genera which fall under bryophytes, let us also have a small brief discussion about their thallus. Amongst bryophytes, the body structure, the body part is called as thallus. Now, let us understand what is a thallus. A thallus is a body organization which is not distinguishable or which is very less demarcated into leaf, stem and roots. Amongst bryophytes, we will see that the differentiation process between roots, stem and leaves are very indistinct. So, let us understand what are different range of thallus organization we could observe in bryophytes. In bryophytes, on the basis of organizational morphology of the gametophyte, they can be segregated into three subgroups: thalloid liverworts, leafy liverworts and mosses. This is a diagram showing thalloid liverwort. A leafy liverwort would look something like this, whereas a moss would look something like this, where it will have its own sporophyte and a gametophyte which is very inconspicuous and smaller as compared to the rest of the sporophytic structure. So, thalloid liverworts are represented by approximately 50 genera and 1,000 of species, which comprises of almost 15% of all liverworts. There are four distinct forms of thalloid liverworts which can be identified. The first one is called as the first one has a dorsiventral thallus, which is very small and delicate, but with a distinct apical cell. They possess simple rhizoids arising out of a central cushion from the ventral surface. In such thalloid body organization, the sex organs are protected by involucres. The genus Spherocarpus and other members of the order Spherocarpales are the representative of these kind of thalloid liverworts. The other type of thalloid liverworts are represented by a dorsiventral thallus, which is deep green and translucent. Though not with much internal differentiation, yet chlorophyllous cells are restricted to layers nearer the dorsal and ventral superficial cells. The parenchymatous tissue is without any air spaces. The rhizoids restricted to the ventral surface are uniform and simple. The sex organs are derived from the apical cell along the midrib on dorsal surface. They are uniform, simple and without involucres around them. These are the Antheridia generally develop earlier than Archegonia in such thallus. They are represented by the members of Mesgariales, which is characterized by the genus Palea. The other type of leafy liverwort, the other type of thallus is represented by the dorsiventral thallus, which are irregular in shape. They grow in rosettes and are of wavy outline. The surface could be very crimped. Every lobe of the thallus possesses an apical cell, the starting point of a new thallus growth. Thalli are typically dark green and without any gloss. Most of these thalli are with extensive internal cavities on the ventral surface and harbor atmospheric nitrogen fixing cyanobacteria. The smooth, simple rhizoids are present on the ventral surface. Only one chloroplast with a pyrenoid per cell is present in the parenchymatous cells. The members of Anthocerotalis, for example, Anthoceros and Notothallus, possess this kind of thallus. The sex organs in these thalli are embedded in the thallus and are endogenous in origin. The other type of thalloid liverwort is one which is thick, dichotomously branched and with prominent apical notch with apical initials. The thallus internally is sharply divided into upper photosynthetic zone and lower storage zone. The dorsal surface possesses epidermis and numerous areolae demarcating air chambers. Each of them open with a barrel-shaped pore. Numerous chlorophyllous filaments arise from the base of such air chambers. The ventral surface has a very thick, prominent midrib. Along midrib arise numerous multicellular dark scales and two kinds of rhizoids, simple and tuberculate. This kind of thallus organization in genera is found in genera Marchantia, Praecia, Conocephalum, along with other members of the order Marchantiales, and probably represent the climax of thallus evolution in liverworts. About 280 genera and more than 7,000 species, comprising almost 85% of liverworts, are leafy liverworts. They resemble mosses at first glance. They grow by an apical cell which resemble an inverted pyramid. The apical cell has 3 cutting faces and a base. This contributes to the axis stem and the laterals, the leaves. The arrangement of leaves is 3-ranked. It is very rarely arranged radially. All the leaves are alike in size. Majority of leaves are 2-lobed and each of the lobe with a growing point established very early in life. Such growth leads to a bilobate cleft leaves at maturity. Of the 3 rows of leaves, 2 rows are placed laterally. The 3rd row of leaves lying along the midventral line are called underleaves or amphigastria. Amphigastria are smaller in size than the lateral leaves. These leaves could be 3 to 4 lobed and with 3 to 4 finger-like projections. The shoots are horizontal and plesiotropic. The rhizoids are produced in large numbers from the side of shoot that is closed to the substratum. In species where shoots are erect, rhizoids are sparsely present. Rhizoids are colorless, brownish when old, or occasionally purple and multicellular. Branching of shoot is very complex and varied. Shoots of this kind exhibit a great deal of variation in gametophytic morphology. Members of order Jungamaniales are typical leafy liverworts. Some prominent genera are Herberta, Lepidosia and Trichocolea. The 3rd type of body organization or thallus organization is found amongst mosses. In mosses, a branched filamentous or plate-like protonymal phase precedes upright, erect gametophores with prominent leaves. The leafy shoot exhibits a great diversity of gross form. The majority of mosses grow by means of a tetrahedral apical cell in the form of an inverted pyramid with 3 cutting faces. The arrangement of leaves on the stem is generally 3-ranked. However, due to growth torsions and consequent displacement, this precise 3-ranked arrangement is disturbed. Branches, if present, arise just below the leaves rather than in their axils. In mosses, the stem and the leaves are referred to as cauloid and phylloids. The gametophores may exhibit vertical growth of several centimeters, bearing laterals only in the form of a subterminal crown of branches, providing a miniature tree appearance, the dendroid habit. For example, in Clamatium dendroides. The cauloid cells may bear prominent conducting strands. In many instances, the leaves surrounding reproductive organs are a little different from vegetative leaves. These are called periacaceal leaves. The rhizoids are branched, multicellular and non-chlorophyllous. Mosses are represented by genera such as Funaria, Priam, Polytrichum, Sphagnum, Andrea, etc. So, having seen various different types of thallus, let us move on to the type study, and we will begin our type study with the genus Marchantia. We shall be discussing morphology, anatomy and reproduction of Marchantia. The systematic position of Marchantia is the division, the genus Marchantia falls under division Bryophyta, class Hepaticopsida, order Marchantiales, family Marchantiaceae. As far as the habitat and distribution of Marchantia is concerned, it is one of the most advanced and highly differentiated member of the family Marchantiaceae. The genus is named after Nicolas Marchant, who was the director of Orleans Botanical Garden, Blois, France. Marchantia is found in moist, shady places near water sources and areas of burnt ground. Marchantia is widely distributed and about 65 species are known from all over the world. In India, approximately 11 species are represented, mostly in the Himalayan regions. The 3 most common species are Marchantia polymorpha, Marchantia nepalensis and Marchantia palmata. Here we are showing a diagram, a picture of Marchantia polymorpha. As far as the habit of Marchantia is concerned, each spore on germination forms a thallus which is dichotomously branched with a distinct apical notch and a prominent midrib. You can always see that various thallus or the body pattern, they will show a very distinct notch where apical cells are present, and we can also see a distinct midrib in the thallus of Marchantia. As compared to Rixia, the thallus is larger, it shows prominent lobes and is better adapted to terrestrial habitat. It generally forms patches and grows extremely well on damp and burnt soils. The green thallus represents the gametophyte phase of the plant. Here in the diagram you can see a profusely growing Marchantia thallus. You can always notice the lobed structure and the dorsal surface. Here you could see white dots. These are nothing but air pores which are present all over the dorsal surface of Marchantia thallus, and the arrow is pointing towards the apical notch which bears apical cell in Marchantia. The gametophytic phase of Marchantia has an external morphology. The adult gametophyte of Marchantia is fleshy, green, dichotomously branched thallus with a distinct median groove. It grows prostrate on the substratum and branches freely by dichotomy. Occasionally, it also takes up a rosette form due to frequent branching, but rosette structure in Marchantia is very rare. The Marchantia thallus is flattened and dorsiventrally differentiated. The dorsal surface shows the presence of distinct rhomboidal or polygonal areas called as areolae with a tiny hole in the center. The pores carry out the gaseous exchange. Each rhomboidal area represents the corresponding air chamber lying below. The dorsal surface also shows the presence of several small cup-like structure called gemma cups. They are generally present along the midrib region. Here in this diagram, you could very well see the presence of these saucer-shaped structures or the cup-shaped structures called as gemma cup. These structures bear small vegetative organ called as gemma, which later on will get detached from the thallus and will give rise to a new young offspring. The position of gemma cup on the thallus, you could see, is primarily on the midrib region. Here also in this you can see they are arranged on the midrib region. The gemma cup has a fringed membranous margin which very well can be observed in the diagrams in this picture also, and they contain numerous gemmae. These gemmae are mostly bilobed structures, and they are the ones which help in vegetative reproduction of Marchantia. Each gemma is green, flat, multicellular body that arises from the bottom of gemma cup. On maturity, the gemmae get detached, fall onto the ground and give rise to two new thalli each from the two notches. Here in this diagram, you could see that there is one notch on the right hand side and the other notch is on the opposite side. So when these gemma fall onto the ground and start germinating, the germination takes place at both the two notches, and thus each gemma gives rise to one thallus towards right and one thallus towards its left, and ultimately they will be giving rise to two vegetative thalli. On the ventral surface, the midrib is represented by a low ridge. It shows the presence of two kinds of appendages, scales and rhizoids. Here in this diagram C, the arrow indicates towards the presence of rhizoids, whereas in the other diagram the arrow indicates the presence of scales. Both of these appendages are found on the ventral surface of the Marchantia thallus. The scales are of two types. One is called as appendiculate scale and the other one is called as spatulate scale. Both type of scales are multicellular, one cell thick and purple due to the presence of anthocyanin pigment, and are arranged in 2 to 4 rows on either side of the midrib. Here is a diagrammatic representation of scales which are present on both the sides in 2 to 4 rows. The rows of appendiculate scales occur near the midrib, therefore termed median, whereas both of the spatulate are found towards the periphery and thus called peripheral scales also. Each median scale arises obliquely from the ventral surface and is divided into a large body and an apical subrotund appendage by a narrow constriction. However, the latter is absent from the peripheral scales, making them appear like a spoon or spatula. Presumably, both type of scales protect from the growing tip from desiccation and retain water through capillary action. Besides scales, the ventral surface also bears numerous elongated multicellular and unbranched rhizoids. The cells of the lower ventral epidermis elongate to form two types of rhizoid, smooth-walled and tuberculate. The smooth-walled rhizoids are thin-walled, slightly larger in and mostly found near the midrib region. They grow directly into the soil and help in the anchorage and absorption. On the other hand, the tuberculate rhizoids are thick-walled, narrow and have peg-like invaginations towards the interior of the cell. They generally originate from under the scales and grow parallel to the soil surface in dense mat. The function of tuberculate rhizoid is to help in water absorption and distribution to the entire thallus. And at maturity, the thalli show several gametophores or gametangiophores, which are upright, stalked, umbrella-shaped structures, and they can be of two kinds, anthridiophores and archegoniophores. In our next lecture, we will see various reproductive structures of Marchantia, the various processes of reproduction in Marchantia, the germination of spores and various other aspects related to Marchantia. Thank you so much.